In this animation, we look at the structure and function of the endoplasmic reticulum, or ER. The ER is a vital compartment or organelle present in all eukaryotic cells and carries out a wide range of functions. It's made up of an interconnected network of branching tubules, vesicles, and cisternae. And on average, the ER makes up approximately 50% of the total membrane surface in an animal cell. The ER is connected to the nuclear envelope, and it's also closely associated with the Golgi apparatus. Based on its appearance under the microscope, the ER can be divided into the rough ER and the smooth ER. So let's look at the rough ER first. So the rough ER is studded with ribosomes on its outer surface, and it plays a key role in the synthesis, folding, and quality control of proteins that are destined for the cell surface or for secretion. Now, in contrast, the smooth ER does not have ribosomes on its surface, and the smooth ER is important for making fatty acids and phospholipids, for assembling lipid bilayers, for metabolizing carbohydrates, and also regulates calcium homeostasis. In the liver, enzymes in the smooth ER metabolize and detoxify drugs and carcinogens. All eukaryotic cells have significant amounts of rough ER, as this is essential to make the proteins needed for the surface of the cell and for secretion. Rough ER is particularly abundant in secretory cell types, such as antibody-producing cells, insulin-secreting cells in the pancreas, or cells of the milk-producing glands, where a large proportion of the cytosol is taken up by rough ER. The ER is a multifunctional dynamic organelle, and it can be influenced by functions both inside the cell and in its outside environment. For example, perturbations in conditions such as the availability of oxygen or glucose, hyperthermia, pH, calcium levels, redox levels, and energy levels can all impact ER function and affect its ability to make and properly fold proteins. Correct folding of proteins is essential for their function. Now, as proteins are translated by ribosomes, they are inserted into the ER in a process called co-translational insertion. Protein folding is a complex process that depends on the interaction of chaperone proteins, foldases, and a range of other enzymes, as well as on an oxidizing environment. Now, conditions that stress the ER can impact its ability to fold proteins, resulting in the accumulation of unfolded or misfolded proteins. This leads to the activation of a specific cellular response called the unfolded protein response, or UPR. And you can learn more about the UPR in our animated video overview of the unfolded protein response.